Thank you. You give very good introductions. I have a very funny story, well, it's tragic funny story about the Lepeak article, but I can't say it until the camera's turned off. But if somebody wants to ask, and you can turn off the camera. You've probably heard at the beginning of lectures like this, you may have even heard it from me, a statement something like the average neuron in the human brain receives 5,000 synapses, or maybe the speaker said 10,000 synapses. So my talk today is in part about the fact that this statement is completely wrong. The correct answer is 5. Right? And the reason for that is the typical cell in the human brain is a cerebellar granule cell. The majority of the cells in our brain are cerebellar granule cells, and they receive 5 synapses. So, I mean, that statement is just to attract you to the fact that there's a different way to compute than what theorists like me often talk about, the cortical way, which does indeed involve many connections per neuron. But there's another way, and I'm going to talk about the other way. It's sort of the cerebellar way to compute. But I'm not going to talk about the cerebellum. I'm going to talk about cerebellar-like circuits. And I think if you know anything about the cerebellum, you'll see as we go along. And there's another surprise about these circuits. First, they use very few synapses, and then they have this other property. So, let me think broadly about a processing system. Typically in neuroscience, we study a lot the input end of sensory systems, for example. We study the retina and the early cortical areas involved in vision. And they're very orderly, they're beautifully laid out, they're very specifically wired. And if you think of them in terms of an engineering term, you would say, well, they're pre-processing the signals in very clever ways, getting ready for us to act upon that sensory representation. If you go to the other end of the system and you ask, how can I move around and control my body accurately, it's also a beautifully organized system of control of motor output that, again, is a specialized system that an engineer would recognize as a controller for some kind of external device. The systems I'm going to talk about have an interesting intermediate stage here. Of course, the intermediate stage is always the interesting one because that's where the sensory gets turned into motor, where decisions are made, inferences are made, and things like that. And the systems I'm going to talk about have a random stage in here. I'm going to try to provide the evidence that there's a random stage and talk about the implications. And this also happens to be the stage where there tend to be very few synapses. So why would you take a system like this and that are so beautifully made on the input side, so beautifully made on the output side, and stick a random phase in between? I think computer scientists would actually recognize right away why you do this. These are called hidden layers in computer science. They're the layers that, again, get you from the input to the output. Now, modern systems like deep learning systems that you've probably all heard about do not use random layers here. They use sophisticated backpropagation algorithms and things like that. But I think most computer scientists would say, well, if you're not going to do that, if you don't have the ability to backpropagate errors and things like that, then putting a random layers in here is actually an extremely good thing to do. It's probably the best thing to do if you can't fine-tune them. Okay, so let me introduce you to those systems. Oh, I have one more thing I wanted to say at this point. Now, if you're going to put a random layer between the input and the output, you're going to have some kind of learning scheme. Because here by random, by the way, I don't mean just unstructured. I really mean different in every individual. So there can't be any universal knowledge here except through learning. And so it's important. I mentioned that we're not going to have learning at this stage, but it's important that we have a learning signal somewhere along the line so we can align this system with reality despite the fact it's gone through a random phase. Okay, so in talking about these systems, I'm going to give some examples and then do some theoretical analysis. And in the case of the examples, I'm really relying on experimental colleagues. I don't do these experiments. I'm going to talk a lot about experimental data. And so I'll introduce you to them. And I just want to acknowledge that I'm a reporter here. I'll tell you when I actually occasionally did something in these collaborations. But mostly I'm just a reporter. Okay, so the first example comes from the fly, comes from the olfactory system of the fly. And that was work work done in Richard Axel's lab, the experimental work I'm going to do by Sophie Caron and Vanessa Ruda in the middle here, and I just, hold the glasses in this collaboration. I guess I'm not the only one. And that's the first part. And then the other part is even a sort of a broader collaboration with a group at Janelia Farm, led by Jerry Rubin. Yoshi Aso is the main researcher. Daisuke is another postdoc of Richard's. And so, I just I'm kind of the fly in this collaboration, the fly on the wall. So I want to acknowledge that. Okay, so let's talk about a fly olfaction. And I'm going to show you that it has the structure that I depicted on that first slide. So instead of having olfaction in the nose, olfaction have appendages like the antenna that are studded with olfactory receptors, and that's their nose. And these receptors, are located in these kind of bristly structures. Like in the mammalian system, each neuron in the periphery expresses one olfactory receptor protein type. There are about 50 of them, and so like in the mammalian system, each cell picks one of them to express and builds those receptors. So here you're seeing a case where through genetic trickery, all the cells expressing a particular receptor have been dyed green. There they are. And now what you can do is follow their axons up into the brain of the fly. They kind of go in the back here so you can't see them, but they come out here and they go to one side on each side called the glomerulus within a structure called the antenna lobe. That's that thing there. So this is like your olfactory bulb, and the same system works in your olfactory bulb, where receptor cells with similar receptor proteins converge onto a glomerular site like this. So this is beautifully organized wiring. If you dyed another set of cells with a different receptor, green, they might go there, others would go there, then there are 50 sites here, each corresponding to a different receptor. So beautifully organized structure. Now, at this point, there's a relay, as in the olfactory bulb, and a cell called a projection neuron, that's what's in yellow here, picks up the signal, and there are about 5 of these for each glomerulus. But if you notice, for example, this guy picks up all its input from one glomerulus. So it's a strict one-to-one relay of this particular olfactory channel. And likewise, there would be other projection neurons that target these other glomeruli. And that carries the signal up to 2 areas of the brain. This one is called the mushroom body, and that's what I'm going to talk about. That's the one that has this structure. And this is called the lateral horn. And that is a structure that is different. And you may just remember, I'm not going to talk about it, but you should remember it. That's responsible for the innate olfactory responses of the fly. So the fact that I'm going to put in randomness, you might think, oh my God, the fly has absolutely no sense of odors, until it learns things. That's not true. It has an innate sense of odors through that system, but I'm not going to talk about that system. So what's the mushroom body? Mushroom body now you can see in red there. Those are neurons that are intrinsic to the mushroom body called Kenyan cells. Their cell bodies are up here. You can see that they receive their input from this projection neuron in an area called the calyx right there. And then their axons kind of make an L-shaped structure that are called the lobes of the antenna of the mushroom body. And that's it. They don't extend out of the mushroom body. So the signal looks like it's hitting a dead end there, but in a little while I'll tell you about the output neurons that are going to pick up that signal and then send it to the rest of the brain for actual action on top of olfaction. But that's the stages we're talking about so far. And the piece of this that I want to talk about is the connections between the projection neurons and the Kenyan cells, this sort of third stage, because I think I should have convinced you in that brief introduction that there's a very orderly single-channel conduction of the signal through the antenna lobe to the Kenyan cells. So then what happens here? And of course, this is where I'm going to argue that the randomness is. So what we're going to look at is the set of connections between these projection neurons and the Kenyan cells. What's the nature of them? You've had this parallel channel of very highly organized olfactory signaling. What happens at this point? Okay, so this was done by a technique in which a photoactivatable version of green fluorescent protein was put in the Kenyan cells. So here you can see a lot of Kenyan cells dimly in green. This is the calyx where they get their input. But then one of them has been photoactivated to be much brighter than the others. And there it is. And you can see, you probably can't see, let me see if I denote it. You probably can't see it, but this process comes down and then branches into what are called claws, and they really look like claws. It's like a hand coming out, and that's the terminal of this guy. It's the postsynaptic terminal, and that claw grabs hold of the presynaptic terminal of one of the projection neurons and makes the connection. Furthermore, although you can't see it here, a bunch of claws from different Kenyan cells all kind of grab hold of that terminal in a structure called a microglomerulus. And that'll be relevant in a second. So the first thing that you can do here from this picture is just count the claws. And that's what I'm showing here. And as promised, there are not very many of them. In this case, it's a little more than the 5 that it would apply for the cerebellum, but it's sort of 6 or 7. They don't get a whole lot of input, these guys, and then I'll come back to that in a second. But my experimental colleagues went on to do a very neat trick, which is to inject a dye into this site, into this microglomerulus, which got taken up then by the presynaptic neuron, by the projection neuron. And you can see that here because there it is. So the projection neuron has taken up the dye, that's where it was injected, and you can see the axon going across the calyx. You can see other terminals that it makes, where it makes connections with other Kenyan cells. So that's fine, but the point of all this is that you can then trace it back all the way back to the starting point, which is the antennal lobe. And then the antennal lobe has been mapped out. It's a highly organized structure. So when you see that this particular guy projected back and got its input, so this is the projection neuron, from this site, you know which channel of olfaction information is going to this particular Kenyan cell. And then you can do it over again by injecting at these different claws and get a whole set of inputs. So you now know that this particular Kenyan cell is getting that set of inputs. So doing this over and over again, you can construct a connectome, a matrix of how the different glomeruli, they're listed across the top here. Those are just their names. It won't matter for us, but these are the different olfactory channels plus some temperature-sensitive channels. There's a little bit of extra stuff. And then here are 200 Kenyan cells. Now, in reality, there are 2,000 Kenyan cells in a fly. So this is not a complete sampling. This is, as many as my colleagues could do before they got tired. But anyway, there's the matrix. Here, a red dot means there was a connection between, this glomerulus and that cell. And the yellow dot means there was a double connection, 2 connections. So, now actually I start to do slightly more than hold the glass. So my job in this collaboration is to say, what's the structure of this matrix? Now you might say, well, it looks pretty random. You've convinced me. But I got to point out, connectomics isn't that easy. It's almost that easy, but it's not that easy. And the reason it's not that easy is that these guys, if you'll note, are organized alphabetically. And these are organized in chronological order of how they were measured. So this matrix is not set up in a sensible way unless the fly organizes its olfactory system in an alphabetical way. So the problem is that, in reality, you should be able to permeate the columns and the rows of this matrix any way you want and then see if it has structure. And let me just show you that this is a scary proposition. So this is just a redrawing of that data in black and white in a little bit different shape. And this is fake data that I made up. So it looks, kind of looks the same. It's not identical, of course, but it looks equally random. But in fact, this fake data has the property that if you rearrange the rows and columns appropriately, it looks like that. It obviously has structure, right? So I mention this not just to say, okay, I solved this problem. But this is the problem of connectomics. Even after you do the segmentation and you get all this beautiful data, you still have to face the fact that the matrix is going to come out in a weird form, probably. And you have to worry, or you have to think about the fact that it may have beautiful structure in it that's hidden by the fact that you scrambled it all up during the data collection process. So there are various ways, I'm not going to talk about them, to convince yourself that the real data, you can't do that, and you can't. I couldn't, and other people couldn't even since then. So the argument is that this is consistent with random draws from a probability distribution, but actually you can probably all see if you run your eyes down the columns, this is not a uniform probability distribution. So all of these guys do not have equal access. But if you're given that probability distribution, there's nothing else in this matrix. It's completely consistent with random draws from a probability distribution. And this has since, there's an earlier paper I'll describe in a second, since been largely confirmed. There's a difference of a couple of cells, but at any rate, in Glenn Turner's lab. And there was an earlier paper that did a clever trick that sort of inferred also random structure. The reason this paper is particularly interesting is that they could actually comment about individual flies. This does not tell you about individual flies because these cells were not all collected in the same fly. But this paper implies that indeed, not only is it random, but it's different in different flies. Okay, so what about the output? Okay, so we've taken it, it's kind of a bizarre situation here. We've taken beautifully organized sensory information and completely scrambled it up in a random way. Seems nuts. Now what about pulling out the information? At this point it's not going anywhere. So here's a picture of one of the output neurons. There are of order 30 output neurons in this system. So if you want the numbers, 50 olfactory channels come in to 2,000 cells. That's a lot of cells for a fly. And then about 20 channels come out. It's more better to say in about 30 neurons. All right, so here's one of them. You can't really see it branching off to other areas of the brain, but it does. But what you can see is that it targets a very specific part of this L-shaped structure. It doesn't go all over the place. And indeed, that is the general theme. So now I'm getting into this very beautiful work that came out of Jerry Rubin's lab at Janelia. So here's a little bit more detailed depiction of these lobes. They're these L-shaped objects. There are actually 5 of them called alpha and beta, alpha prime and beta prime, and gamma, which would sit right here. It's been peeled away. So the L-shaped structure is sort of these 5 structures, and they are broken up, or if you want 3 structures, if you count alpha, beta together, they are broken each up into 5 pieces in the way I've depicted here. That was earlier work from Ito's lab. And it turns out that the output system obeys this compartmentalization extremely rigidly. So if I kind of figuratively depict the mushroom body this way, here's the part I was talking about. These arrows are supposed to be the projection neurons coming in. This would be the calyx, and this is obviously that L-shaped structure. Then if you look at the different output neurons, they lay onto this within these compartments very strictly. So their dendrites, where they pick up the signal from the Kenyan cells, will not cross these boundaries, and they will lie in this structure. Furthermore, I'm trying just to get the idea that after randomness, a strict order is now being posed on this system. I forgot I'd had this one. So here you can see with labeling in different colors of the different output neurons, how they just sit in non-overlapping compartments along these mushroom body lobes. You're seeing both sides here. Okay, so the other thing is that if you look at the transmitters used by these output neurons, again, you see that it's laid out in a very orderly manner within this structure. And then, so the idea, I just put these arrows in just to remind you, these blocks correspond to the dendritic fields of neurons that will then carry signals out into the rest of the brain. So this is how the olfactory information moves along the pathway with this highly structured system imposed. Now, there's more to this structure, and if you remember in my first diagram, I had the structure, randomness, structure we've gotten there was a learning signal. And the learning signal in this case is a dopamine signal, and there are a set of dopamine neurons that also strictly target these compartments, single compartments like this. You don't see them crossing the line. So there's a very specialized learning signal being applied individually to these output channels. And here you can see the beautiful alignment between the dopamine innervation and the signal being pulled out by these different output neurons. Okay, and furthermore, that dopamine system is also laid out in a very beautiful structured manner. The compartments in the kind of the vertical part of this thing are associated with dopamine signals that are activated by punishment. So this is a very simplistic division of dopamine into kind of good and bad, but that's what this is. And then the reward dopamine signals are along this vertical lobe. Horizontal medial lobe. Okay, so you have this beautiful structure, and let me just quickly sort of say how it's generally thought to work. This is old ideas due to Heisenberg, for example, and it kind of goes like this. This, in fact, is the Heisenberg version, which I'll update a little bit. So the idea would be like this. An odor comes in, and it activates a certain subset of these Kenyan cells through the random connections. So the fact that I drew these two lines as black, meaning they're activated, is in one fly. In another fly, it would be another set of lines. Okay, now if you imagine that, let's say you reward the fly. I guess this was the reward-based part. You give the fly some sugar right after that. Then a dopamine signal gets sent to this compartment, and it modifies, the hypothesis is it modifies the synapses. Actually, there's evidence for this. It modifies the synapses of these recently active pathways. And I've drawn it as strengthening, which is probably what you would mostly guess. And the idea then, the dopamine signal goes away, the fly merrily goes on its way, but now the next time that odor comes in, it hits these altered synapses, and the fly gets a signal predicting the sugar reward, something like that. That's the idea how it works. Now, the only thing wrong with this picture is that it's probably backwards. It's cerebellum-like in the sense that everything is backwards. In the sense that it's probably more likely that this output neuron responds to all odors and it fails to respond to this odor. It's backwards. So these synapses actually get weakened, not strengthened. It was just easier to draw this way. Okay, so I hope I've convinced you then that there's a front end on here that's highly structured. Due to beautiful work of Rachel Wilson, we actually know that this system is doing very nice signal processing things, like averaging signals, normalizing signals, things that you would do before doing the heavy-duty cognitive stuff. I hope I've convinced you that it's a random connection in there, and then, of course, that there's a very highly structured output, and that there's a learning signal that is also very highly structured that allows this system to draw information out of it. Okay, now, why would you build a system like this? There's an old quote from really the person, I think, who discovered mushroom bodies, which is, very nice because it's sort of the origin of free will in flies. I know there's philosophers, around, so I don't know about humans, but flies, we got it. We've located free will. But really, this does mean that a fly, which is really a pretty hardwired animal, if you look at many of the neurons that at Genelia and other places, there are now genetic lines that identify small families of neurons and allow you to look at them. They look the same in every single fly. And yet here you have a system that we're arguing is different in every fly. So there's a little individuality in the fly, and that allows each fly to learn from its experiences. It allows flies to be different, and perhaps even gives them free will. Okay, so example number 2, and then I'll get into the theory. Example number 2 is a circuit in the fish, in an electric fish. Here, the experimental heroes are on the top here. In particular, Nate Sawtelle, my colleague at Columbia, did the bulk of these measurements. He kind of said, hey, I've got all this data. What am I going to do with it? And I just kind of let it be known that that data was around. And 2 theory students and a half Anne and Greg and Patrick just jumped on it and took care of it. You know, I like to promote a very free interchange of people, and so that really happened here because these Anne and Greg worked with me, but Patrick actually worked with another junior experimental professor, not Nate. And Attila Lassanchi, who should get a lot of credit for kind of giving him the freedom for Patrick to work on this project, as well as writing about 18 papers with Attila. Okay, so what's the deal with the fish? Many fish, including this one, but including fish that we do not call electric fish, for example, sharks, have very sensitive passive electroreceptors on their bodies. And these receptors are used to detect electrical signals caused by the muscles of various prey in the water, or in case of sharks, underneath sand where you can't see them, or in murky or dark water. And so a fish can sense that electric field, know there's something out there, and go eat it. That's the idea. Now, the special thing about electric fish is they have a second system. It's important to realize we're talking about two different electrosensory systems here. The other system is an active system in which they have a specialized organ. It's a modified muscle that can generate an electric field. And that electric field then, you know, it goes out into the water, and depending on the positions and the structure of objects in the water, the fish then picks up that active signal through a separate electrosensory system, and do, you know, as it senses distortions, it can figure out, oh, there's something out there. Now, so the pulse is a very short and very strong electric pulse called an electric organ discharge, EOD, you'll see on my slides. And that, of course, is the whole meat of this active system. It's like the chirp of a bat, if you want. It's the thing that makes it work. But for the passive system, this is a complete disaster, because this field is about 1,000 times bigger than the fields that the system's trying to sense. And furthermore, because these receptors are so sensitive, they get sent off into this big ringing pattern like this, by the fish's own electric discharge, which is not an interesting signal. It's not a bug in the water. Furthermore, you know, if you notice, this lasts for about a couple hundred milliseconds, and even when the fish is kind of relaxed, it typically is firing these things every 200 milliseconds. So if you didn't do something about this, all the passive system would tell you about is your own electric field and not about the interesting stuff. So the system I'm going to talk about is another one of these cerebellar-like systems that cancels this predictive field and leaves, you know, a possibility of sensing this much weaker field. So the idea is the fish knows that it's discharged its organ, and through experience, it should be able to predict this pattern of input and cancel it out, and then look for deviations from that would indicate this bug. So that's the way the system is designed. And really, I'm getting most of the information I'll give you was done by Curtis Bell over his entire career. He and his lab really unveiled many of the properties of these things. Nate and the theory students that I mentioned, we just got in for a last step, but it was an important step. Furthermore, Patrick Roberts really did some incredibly beautiful models of this system, and again, we just kind of put in the last step in that modeling structure. Now, just to make you realize how hard this problem is, although I'm not going to talk about this too much, these are plots Ann Kennedy made, and then Tim Reckworth did some measurements in Nate's lab, about the fact that if the fish, when it's doing this and has to cancel its field, it's probably going to swim along. It's probably going to be bending its tail. Now, when it bends its tail, the electric field is going to get stronger and weaker on one side or the other. And therefore, when the fish cancels it out, it has to not only know that it made a discharge, it has to know the shape of its body as well. So all of that information has to be integrated, computed, and a prediction of these fields that you're seeing in these diagrams at the surface of the body has to be made so that the fish cancels that out. So here's how the system works. It's the electrosensory lobe of the fish, which is kind of an offshoot of the cerebellum, or it's very similar to the cerebellum and has a similar developmental origin. And it looks like this. Into this structure become things called mossy fibers, which if you know the cerebellar terminology is also what the inputs to the cerebellum are called. And those carry the information needed for this computation. If you want to know what field you're going to produce yourself, if you're a fish, you have to know that you fired the electric organ. That's what's called this corollary discharge signal. And you have to know that, you know, your tail's bent a certain way. So this is this body configuration. And that's known to be represented on these input fibers. Okay, now I should just mention that you know, we have systems like this too. You know, I kind of tend to wander back and forth when I talk, and you know that you guys aren't doing the wandering, it's me, because my visual system is getting the motor things. It's saying, you know, you're about to move right, so all the people are going to move left. My visual system knows that through a corollary discharge system. This is very much like that. In fact, since the electric organ is a muscle, it's really quite analogous. You want to remove the effects of your own body, so if you guys all do get up and run out of the room, I know that's you and that's not me. It might be me, but in a different sense. Okay, so now the calculation that we're going to talk about is done, or thought to be done at any rate, in a cell called the median ganglion cell, and that's like a Purkinje cell of the cerebellum. And that's like these output neurons of the mushroom body, okay? That's the output system for this. And in the middle are, like in the cerebellum, a lot of granule cells. These granule cells get a small number of inputs. In this system, it's about 4 from the mushroom bodies. Come down, make a parallel fiber system, and make about 20,000 synapses onto these cells. So it's not 200,000 like a Purkinje cell, but it's still a lot. And I should have mentioned, this parallel fiber system is very much part of this action. If you noted in the mushroom body, that L-shaped structure was a parallel fiber system. It was the axons of the Kenyan cells in parallel tracts. So they all have parallel fiber systems. Okay, so the work from Curtis Bell's lab actually worked, there was the slice work, so that we actually know what the plasticity mechanism is here. It's a spike timing dependent kind of mechanism, and it really does explain the whole calculation. And we also know the nature of the error signal, and the error signal is a little bit weird in this case. So the error signal is actually the sensory input. Because the idea is that if this cell responds to this repeated pattern, then it's making a mistake, right? I'm going to assume that most of the time there isn't a bug out there. And so it is response to the sensory stimulus that tells you. If you're repeatedly responding in conjunction with the corollary discharge, that's an error signal, and you have to subtract that out. And in fact, what this system does is that it gets this sensory ringing input here, that's coming into the cell, and from this whole business, it generates a prediction which is called a negative image. So it's a negative prediction. It predicts that it's going to get this, and when you add those two up, the idea is you should get 0. I'll show you actually that in a second. So that's how the system works. What I want to argue in this system, by a little different argument than I did before, is that this stage is once again random. So we have, well, you know, I'll go through this again, that this stage is random. But we did it in a different way. With the fly, I gave you some anatomy to argue that it was random. In this case, it's physiology compared with modeling. And the way we did that is the following. Nate, in the lab, measured the responses of about 200 granule cells. There's a common number in this experiment. If you notice, I had 200 cells in the fly and 200 granule cells, which is experimental measure 200 cells, and then they quit. That's the theorem. And that's good for theorists, because if you remember, there were 20,000 inputs here. So after they get exhausted with 200, they call in the theorists and say, model the rest. I can't take it anymore. And that's kind of what happened here. Okay, so we had the job of generating from the data 20,000 granule cells, model granule cells. And we did it. And we did it under the assumption that these inputs were random. In other words, we took Nate's recorded mossy fiber data, we randomly sampled it, and we built a bunch of granule cells. Okay? Then what we did was to take randomly 200 of those and pull them out and order them by an algorithm that Nate had used for his data, and we got this. So this is the model, You're looking at normalized voltage fluctuations of the granule cells following the electric organ discharge command. So you see various things. You see pauses, you see late responses, you see early responses. The point is that the cells that Nate measured look like this. And this is not, these cells are not a model of these cells. They were just randomly selected cells ordered in the same way. And presumably, Nate's cells were also randomly selected by, wherever his electrode happened to hit. But that's the evidence I will give you that this system is randomly wired. If you make that hypothesis, you get a pretty good model of the data. Okay, so again, this system has this general structure, a highly structured input, a random, highly expanded representation in the middle, a learned output, and an error signal. And in this case, you even know the plasticity mechanism. All right, so now I just want to talk, I think I have time before I do the theory part, I just want to talk about some really cool experiments that are not central to my message, but they're really cool, so I'm going to talk about them. And they were done in Nate's lab, so all credit should go to Nate. I'm the rapporteur here. And that's because, after we did this work explaining, if you want, the cancellation, it started to worry about, okay, how does the fish actually detect the bug, though? So in this system, there's kind of a cool thing that happens. When these experiments are done, the fish is paralyzed, so it doesn't wiggle around, and then, electrodes are put in and all that. But when you paralyze the fish, you get rid of this signal because that was made by a muscle, and the muscles don't, it's a modified muscle, but it doesn't work in that case. So this signal, which is the normal act of the discharge, has to be replaced by a mimic. And what Nate does is to record the discharge that the fish is making on a nerve, and then instead of this natural field happening, you just create a field in the water that whatever you want, but you link it. So every time the fish thinks it's discharging its electric organ, you blast it with this. And that's kind of cool because now you can play all sorts of games. So let me look, I'm going to show you the output in the case. This is experiments that had been done by Curtis Bell, but this is just Nate's version of it. So when you start this experiment, you haven't turned on the field yet. The fish has been firing its organ without any electric field at all, and it gets used to that. So when you first turn on the mimic, first of all, also the mimic doesn't really perfectly represent the fish's own field. It's a weird field. So at first, these neurons at the output respond very strongly to this electric organ discharge. That's the fish responding to its own field. That's not good. But if you wait a while, minutes, this system canceled that out. So here is the same cell, and now it's completely not responding to the mimic. Now you can see that what's happened by then just failing to fire the electric field one time. So you just basically turn off the electric organ discharge. The fish makes a command, and now you see this strong inhibition of the output, which is what's canceling this. So that is the negative image that is formed in response to this electric field. So that's step one. The system really does work like it does, and I have to say the model works exactly like that. The model works beautifully. Okay, now what happens if you add a bug? Now the bug is also a mimic. The bug is a little wiggle of an weak electric field that kind of looks like a bug, hopefully, to the fish. You'll see that it actually does in a second. Okay, and this, as I said, was done by Armin and Ella in Nate's lab. So now what we can do is we can record from those output neurons and do a kind of ROC analysis. We can say, from that recording, can we tell if the bug was turned on or turned off on that trial? And what we find is, in this particular cell, we could almost 80% reliably tell that the bug was out there from the output neuron. Now, that was without any electric field, any discharge. The bug's electric field was there. Now you turn on this artificial discharge, and the hypothesis is that makes it hard to detect the bug. And indeed, it does. So now the detection ability from this same neuron goes down to almost a chance. And of course, that's because it's making this huge response to the self-generated field. And that's bad. But as I mentioned, over time, the fish learns to get rid of this and and the detection goes back up to 80%. If you add that electric field, but you don't couple it to the nerve signal so that the fish cannot predict it, then the fish can never learn much above chance, or this circuit can never learn. So the circuit also works like, Curtis Bell always said it did. But this is the coolest experiment. The fish is sitting there paralyzed, but nevertheless, it can tell you that it sees the bug. And the reason, the way it does this is it gets excited because something's out there, and it increases the rate of its electric discharge. So it's going there, at 5 hertz. It senses the bug, and it suddenly increases the rate of discharge, in natural conditions in preparation to really localize that bug and eat it. So here you see an example of that. So here is the prey-like electric field, and you can see that the fish upped its rate, and therefore the fish is telling you, I saw it. Now you can do the same thing with the fish. So here's an example where this response, what we're measuring is the z-score of this response, reliably occurs in response to the fake bug when the self-generated field has been turned off. Now you turn on the self-generated field, and as suspected, you go back to the fish cannot detect the bug anymore. Gradually that returns and comes back to a good level. And again, if you discharge this field and you don't let the fish learn about it by decoupling it to the signal, the fish can't learn to detect the field. So this system works just like it's supposed to work. Okay, so now I get to be a theorist at the end. So this is work that was started by Cameron Harris and Chaim Sampolinsky actually at the MBL computational course, and then Ashok Kumar Litwin, who is a postdoc working with me, and I kind of have continued this work and continued working with Cameron and Chaim. So this is a general analysis, if you want, of systems like this, and we're particularly addressing why are there so few synapses in these systems. So the idea is the following. Suppose you have M channels of information coming into a system. So in the fly, where we know the most about this, these are the 50 olfactory channels coming in. And then you hook them up through capital K synapses. So try to remember the letters, even though they're not a particularly great choice of letters. So here, for example, 4 synapses. You hook them up to these granule/Kenyan cells, these intermediate cells, and that these connections are made randomly, okay? You choose one of these. We got another one of these. It's kind of dying. You randomly choose one of the inputs, and you hook it up. Okay, and then you repeat that over and over n times. And in the case of the fly, that was a couple of thousand. You got another one? Good. Yeah, thanks. Okay, and then the idea was those would go to an output neuron, and great things would happen. But what we're interested in here in particular is why did nature choose to make so few connections onto this intermediate layer? I kind of argued for random, but why so few? In computer science, they would typically understand, build a random layer, but they would connect all of these to these guys. They wouldn't do it this way. Okay, so why are those numbers so low? So the first thing I want to do is just a little combinatoric calculation with you. So the idea here is that this intermediate layer is set up to be extremely diverse. What you want to do is take those 50 channels and expand their dimension as much as possible by making all sorts of different combinations of inputs, that are added up in these cells in many different ways, getting lots of diversity in the response, and then the output neuron can look at all of that and make some inference about it, which is the whole point of this system. So we want to maximize diversity, and that means we don't want to keep making the same cell over and over again. These yellow cells, when I put up n of them, you want them all to be different, because diversity is the name of the game. Things like, averaging to reduce signal noise, that's already been done. We're in a different regime now. All right, so how many different ways can you make a granule cell or a Kenyan cell? Well, if you have m inputs and you choose k of them, then it's m choose k is the number, the binomial coefficient. So you know how many they are. Now, I call this B for a very good reason, because we want to know what's the probability that if you randomly start assigning n cells in this way, 2 of them will come out the same. That's the same problem as the birthday problem. Birthday problem is that I ask people in this room, what their birthday is, and it's almost guaranteed for a room this size that 2 of you will have the same birthday. Okay, it's sometimes called the birthday paradox because people think in their heads you need 300 people in the room before there's a high probability, but you need the square root of 300 people in the room before there's a high probability. Okay, so it's the birthday problem, which means the other advantage, you can look up on Wikipedia what the answer is. So not that I advise that for the students. You can also calculate it if you have a good day. Okay, so the probability that they're all different is just given by this quantity in terms of the B. B is like the birthdays, but for cells, it's their identity in terms of who they receive. And so now you can plot this for the fly. And what you can see is, if you don't have enough connections, it's inevitable. You can't make 2,000 different cells from 50 inputs with one input per, you can only make 50 different cells with one input. But then it rapidly rises, and pretty soon it's very close to 1 that they'll all be different. And in fact, there's where the fly lives. The fly tends, in all these calculations we find, the fly likes to live kind of at 95%. 95% probability that they'll all be different. It's willing to give up on the last 5%. So that's the first argument, why so few. There's just no point in making more. They're already all different with a, with a high probability. Okay, now I mentioned that the point of this representation is to be high dimensional. And by that I really mean that, the data cloud that was in 50 dimensions originally has been sort of thrown all over the place into a very complex, it's still a 50-dimensional surface, but it's curved and windy, and it sort of occupies as much dimension as possible. And there's a formula that you can use. I'm not going to bother to go over it, but there's a formula for this dimension, which I'm going to use. And I'm going to make a note of a fact. Here is where the computer scientists might say, I don't understand why you're getting this result. The reason we're getting a result is because synapses are excitatory. So we're going to put in the constraint that in all of these systems I've mentioned, the synapses coming in are excitatory, not inhibitory. Now, if you want to ask in the questions what happens if you add inhibition, I'm willing to answer, but I probably won't do it now. Okay, so this was the result. Never mind. Never mind. I'll just say it in words. So this is the plot for the dimensionality as a function of the number of synapses in this system. Goes up, goes down. And again, the fly sits there very close to the maximum. Now, why does it go down? The reason it goes down is that if you have positive vectors, let's say we're in 3 dimensions, and you try to jam more of them in, those are the weights of these things, they tend to get more and more together. That's what those little calculations that I didn't bother to show was. So what's happening here is, if you remember, the diversity went up and then just saturated, so that you're not really getting any more diverse, but the weights are being jammed together, and here you're getting bad correlations. That's what's happening here. That's why things are going downhill. Okay, so what else would you build these systems for? Whenever anybody has 3 reasons for what they're telling you, it means that they don't know what the right reason is. Otherwise, they would just tell you the one. So that's true here. The problem is we don't really know what the fly does with its mushroom body exactly. So it seems like dimension's a good idea, but how do you know? Here's another thing. This sounds somewhat more reasonable. This is the XOR problem, which is very famous from computer science. Namely, the idea is, what if you want to, let's say, respond to a stimulus X, respond to a stimulus Y, let's say you're afraid of both of them, but when it's X plus Y, it's fine, it's safe. That's a little hard to do because, if you just had a threshold and X was across threshold and Y was across threshold, then X plus Y would be even more across threshold. So this is, a little bit of a tricky calculation, and it's known that hidden layers like this are really good at solving this problem. So we decided to do a little experiment about the quality of XOR in a system like this. So the idea is you look at the response to stimulus 1, or I guess I called it X. You looked at the response to stimulus 2, that's Y. And then you want the response to X plus Y, or 1 plus 2, to have as big an orthogonal component to it as possible. Because if it lies in this same space, in the space spanned by 1 and 2, then you can't solve the XOR problem. So the measure I'm going to talk about is sort of the length of this projection relative to the lengths of those projections. And it has the same property. Goes up, gets to very close to a maximum. Again, the fly is pretty close to the maximum, goes back down. So for solving these problems, having 5 or 6 or 7 synapses is great. The last calculation is a sort of a classic perceptron. If you have a computer science background, that's what computer scientists like to do with systems like this. They like to have this output unit categorize stimuli, let's say into 2 categories, good and bad, something like that. And so you can calculate the noise problems, sort of effects on a system like this. And here's where you pay a price for randomness. If you want to, why didn't computer scientists just keep building random systems? I'm not sure they knew this, but I think this is the reason. That is, and it's a very nice calculation done by Bakhtash Babadi and Haim Sampolinsky. And it concerns the sort of noise in this system. So the sensory representation, that's the M lines coming in here. And there's going to be noise there, but I like to think of it as really environmental noise. Namely, if you smell an odor and you know it means fire, the next time you smell it, it's not going to be identical. It's going to be a little different, but you have to recognize, ooh, that's still a smoke smell. I better run out of here. And so, there's a natural variability in sensory stimuli that we want to call the same. And the problem with random representations, it does this beautiful dimensional expansion. That's the good news, is that it exaggerates the noise. And that's what this plot is saying. The noise in this mixed representation, that's the granule cells, the Kenyan cells, is bigger than the diagonal here. It gets expanded. Now, Bakhtash and Haim did this calculation assuming complete connectivity. In other words, all M inputs go to all cells. And what Cameron realized that got this whole project started is that if you sparse in that, this problem gets less severe. So you have noise advantages in cutting down on the number of inputs. And again, you find if you do sort of a signal-to-noise ratio that the fly, in fact, lives pretty close to the minimum of the signal-to-noise ratio. Okay, so I'm going to wrap up by just saying one thing. I had more, but I'll stop. And that is that, okay, so I started off saying that the average number is 5, but you might point out is, there's lots of the brain where the average number is not 5, it's 5,000 or 10,000, whatever. So what's different about cortex? I've been talking about cerebellar systems, and the one hypothesis is, I've been talking about systems in which on this middle layer, you do not do serious learning. Now, in fact, no learning in these models. You do all the learning at the output. That is the easiest way to learn, right? Then you don't need backprop and all that. So that's the easiest way to learn in computer science. It's the easiest way to learn probably in biology. But it costs you, and in a sense, cerebella have followed one path, saying, okay, we're not going to learn in that layer. We're just going to make it gigantic, and we're going to cut down on the number of synapses, and that's a good plan. But suppose you do allow learning in that layer. What you find is it flips. And now you suddenly benefit from lots of synapses. It's kind of intuitively obvious, but it comes out of the calculations too. So what I would argue is that the systems I've been talking about are systems based on this structure. In other words, evolution figured out some way to teach the output. And as soon as that happened, there was huge pressure to expand the size of this system, to cut down on the number of synapses, and build the best randomized system you can. And that, ultimately the huge the case of that is the cerebellum with a zillion granule cells and these output neurons with huge numbers of inputs and a sophisticated learning system. And then you have the other pathway, which are systems that figured out how to do the learning in this layer in the same way as deep mind figured out, and backprop people figured out how to do it. And then the evolutionary stress is the other way. Now increase the number of synapses here, make a more equal structure so the output neurons are not so freakishly different than the input neurons, and build a cortical-like system. All right, I'll stop there and take any questions.